Okay, so why don't we go ahead and get started? It is my distinct pleasure to introduce to everyone today Dr. Peter Sacheri. Distinct for many reasons. One is that Peter and I have known each other for many years. We've been meeting up for over the past 20 years at the American Society of Human Genetics Conference. And it's been fun to watch him progress from graduate school and on to being a full professor at Case Western. So just for a little bit of background, he did his undergraduate work at Gettysburg College and got a BS in biology. He then went on to do his graduate work in biochemistry and molecular genetics in Eric Hoffman's lab at University of Pittsburgh. And he then went on to do a postdoc at NHGRI in Francis Collins' lab. Then he moved on to Case Western University. As I said, he rose through the ranks. He is currently the Gertrude Donnelly Hess Professor of Oncology, as well as Professor of the Department of Genetics and Genome Science. So today he's going to talk to us about interrogating the enhancer epigenome to deconvolute complex diseases. Peter, welcome.
All right. Thank you, Reed. Whoa. Sorry about that.
That's a good question. Let's see my volume. All right, try now.
Hello. Hello. Can you guys hear me? Okay. All right. Well, I'm going to start by thanking this guy, Reed, for the invitation. As he said, we've known each other for 20 years. Life is always a lot more fun with Reed around, as you can tell from this picture. We're drinking fabulous $20 pina coladas at an island off the coast of San Diego. So if you haven't done that, I would encourage you to do it. Again, Reed's a great colleague and friend. So thanks for the invitation, Reed. So my lab, we study the epigenome and we focus on gene enhancer elements. Okay, so as all of you probably are very well aware, DNA holds the instructions. Okay, but the epigenome we really think of as the architect, or more specifically the foreman. Okay, it directs actions like turning genes on and off. Okay, we all know a human being has trillions of cells, all specialized for different functions: bone, heart, muscle, skin, so on and so forth. And all these cells carry virtually the same DNA. What's different about those cells are their epigenome. And their epigenomes dictate very specific gene expression programs that define those specific cell types. And an analogy I like to use for enhancer elements is shown here. We like to think of them as almost like dimmer switches connected to a gene. And there's often multiple enhancer elements that control the gene's expression. Obviously, it's way more complicated than that in the cell. It's a gross oversimplification. And what do we know about gene enhancer elements so far? So if we zoom in on them in the context of chromatin and DNA wrapped histones, first we can deduce that they're often located in open regions of chromatin, and these histones that flank enhancers tend to be covalently modified with very specific modifications, namely the K4 monomethylation mark and histone H3. And active enhancer elements are also containing the H3K27 acetylation on these histones. Enhancers can be bound by a variety of transcription factors. Some of those are shown here: P300, BRD4, mediator. Lots of other factors can assemble at enhancer elements. Enhancer elements, just like genes, are transcribed by RNA pol II, and they make short little eRNAs or enhancer RNAs. And they're thought to act on their target genes through looping of chromatin, through physical interactions dictated by higher dimensional chromatin folding. And what we've also learned is that gene enhancer interactions tend to be constrained within these topologically associated domains, or TADs for short. And I'll be touching on some of these concepts as I go through my talk. And we've also learned that there's a growing list of genetic diseases that are caused by mutations that disrupt enhancer gene control. Here's both in trans and in cis, and here's some examples of those. So Kabuki syndrome, this little kid on the left, this is normally caused by mutations in the MLL MLL is a methyltransferase, and it's specifically responsible for depositing that K4 monomethyl mark at enhancers in the genome. And so this would be considered enhanceropathy due to a mutation in a gene in trans. Perhaps the most famous enhanceropathy is preaxial polydactyly. So this is actually driven by a mutation that's residing in cis in the enhancer of the sonic hedgehog gene. And what that mutation does is it moves with the expression of the sonic hedgehog gene in the developing limb bud. Okay, and the result of that messing with the timing and the expression of the sonic hedgehog gene results in the formation of this extra digit. So these kids are born with multiple fingers. And we're really starting to learn that it's not just these very rare diseases that are caused by enhancer malfunction, but much more common diseases. Okay, the things that you and I will probably eventually get. Okay, heart disease, diabetes, cancer. Okay, a lot of these are, or at least predisposition to these disorders is due to DNA variants that reside within enhancer elements and probably alter the expression of the genes that are important for these diseases to manifest. I'll point out that these are just variants. They're no way causative of these diseases. They merely provide a predisposition. There's something else in the environment that's usually required for these common diseases to manifest. Okay, so back in 2012, my lab was amongst the first to demonstrate that there's widespread alterations in alterations in enhancers in cancer. And we did this through a lot of ChIP-seq profiling of enhancer histone marks, particularly in colon cancer. And what we had noticed is that in colon cancer, when you map these enhancer histone marks, you find that there's regions where you see gains of acetylation or monomethylation corresponding to a gain in activity of the enhancer. We call these VELs, variant enhancer loci, and there's gains all over the place, and there's losses all over the place. And as you might expect, these losses of these enhancer histone marks generally correlated with decreases in associated gene expression, and gains in the enhancer histone mark correlated with increases in the expression of related genes. And this might have been somewhat predictable, but a lot of the genes that we were seeing were really signature genes of colon cancer. And so what it told us was that the change from a normal cell to a malignant one is driven fundamentally by oncogenes, but oncogenes in cooperation with these changes in the enhancer epigenome that are really driving a state change, very much like the state changes that occur during development. Okay. And so this discovery back in 2012 was really served as a launch pad for a variety of basic and translationally focused research projects that are going on in my lab. Okay, we're homing in on the mechanism by which this enhancer activation and deactivation can happen. Sometimes there are mutational mechanisms involved, other times there's epigenetic mechanisms. We're doing a lot of profiling studies in a variety of different cancers because it turns out you can use the enhancer epigenome to subtype cancers very well and find subtypes that you wouldn't normally find through either gene expression profiling or mutational profiling. We've been doing a lot of work in metastasis. Okay, we've known for a while that when you sequence primary and metastatic tumors, you usually don't see too many differences in the mutational landscapes. In a lot of cases, they're virtually identical. Okay, now we have some evidence that what's actually happening in metastasis is one of these state changes driven largely by changes in the enhancer epigenome, and you can block those changes or prevent the activation of those genes and actually prevent these metastases from forming. The evolution of drug resistance, okay, there are clearly mutational mechanisms that can drive the emergence of drug-resistant phenotypes, but there's also epigenetic mechanisms, and in particular, changes that happen to the enhancer landscape that can drive the formation of a resistant cell type. And as I mentioned, we've learned a lot about how SNPs can alter enhancers and confer predisposition to a variety of common genetic diseases. And lastly, we're focused a lot on how you ultimately leverage all of this basic science for translational work, like how do you get to the clinic? So part of my lab is focusing on how you drug the enhancer epigenome, particularly in cancer, and try and make headway clinically. So the first main story I wanted to tell you about started with an MD PhD student in my lab named Andrew Morton, who started thinking about the various mechanisms by which oncogenes are activated in cancer cells. Okay, we've known about these mechanisms for decades. And we've known that, of course, you can have activating mutations that switch on an oncogene, probably the most famous being KRAS G12D mutation that activates KRAS. Translocations can create fusion proteins. Those fusion proteins can be oncogenic. As I mentioned, like we know you can have these VELs that get turned on that can lead to variant transcription activation. But another common mechanism that we hadn't dug into at the time was amplification, gene amplification. Okay, we know that proto-oncogenes can be converted to oncogenes simply by, not simple, but by massive amplification of those regions. And so Andrew set out to determine, well, is there any role for enhancers on these oncogene amplifications? Okay, is it just the oncogene that's amplified, or is it pulling along neighboring enhancer sequences that could be important for activation of the oncogene? And he wound up publishing this cell paper, and I'll just get to the punchline. And what he showed was that there are functional enhancers on these oncogene amplifications. But a lot of times when this happens, there are these extrachromosomal circular amplicons that I'll tell you more about. And this was a collaboration with Jeremy Rich's group at UCSD, Matthew Lupien at Princess Margaret Cancer Center, Richard Solari, a really talented informatician who founded Axiatal, and folks from my lab. And I'll also say that this has been a very hot area. So when we published this paper in Cell, Paul Michel's group published a very similar finding in Nature. And then less than a month later, Anton Henson's group published a nice paper in Nature Genetics showing something similar. And so this field, this was in the end of 2019, and it just exploded here, and it's been exploding since. Okay, so back to the story. So back to oncogene amplifications in solid tumors. So the way to think about this, about oncogene amplifications, is that they really come in different flavors or classes. Okay, there's low-level amplification where you may just have 3 or 4 copies of a large region or even a chromosome amplified. But that's really distinct from high-level amplification, where you have multiple copies of a segment of a DNA or a locus that's amplified. And this can occur in at least 2 main ways. One way is this breakage fusion bridge mechanism that Barbara McClintock described back in the 1950s. And another way that you can get these large focal amplifications is through the production of these circular extrachromosomal DNAs. Okay, so these are circular structures that are about 0.5 megabase to 2 megabases in size. Okay, so they're big, but they're not nearly the size of a chromosome. So again, Andrew said, well, what else besides oncogenes could be on these amplicons? And he decided to investigate this question in glioblastoma, where we know EGFR amplification is one of the most common driver events for glioblastoma. And often these amplifications arise through this mechanism called chromothripsis. This is a chromosome shattering mechanism where the pieces are kind of put back together at random. And it's also known in this cancer that you often have these circular amplicons containing EGFR on them. And so Andrew reasoned that we could just look at patient data and determine whether or not there's evidence for enhancer selection on amplicons. So the way he thought about this, he said, okay, if we take patient samples and we look at just look at the width of the amplicon in each patient sample, if all the amplicons are just centered about the oncogene kind of randomly, and the minimal region of amplification always contains the oncogene, then there's selection for the oncogene and really nothing else. But on the contrary, if these amplicons are skewed, either 5' or 3', it may towards a non-coding region, it may suggest that there's something in that non-coding region that's under selection with the oncogene. So here's the data. This is a busy slide, so I'll walk you through. But this is, I think, 150 different amplicons from patients that were evaluated. So the red bars correspond to the width of the amplicon, and he's got them all lined up to the EGFR locus. And you can see that virtually all of them span the EGFR gene, but there appears to be this skewing upstream. Looks like it's pulling along this Sec61 gene, but actually most of them, the minimum region of application is all this non-coding stuff right here. It turns out, if you look at the ChIP-seq data mapping what's there, looking for enhancers, sure enough, there are two enhancers, which we call the EGFR enhancer 1 and enhancer 2 within this region of amplification. These regions show near perfect co-amplification with the oncogene in almost every instance. We wanted to validate that these were in fact enhancers regulating the EGFR gene. So we used CRISPR-Cas, CRISPR-Cas9 crab fusion proteins, where we silenced the EGFR enhancers and the promoter, and sure enough showed that dropped the expression of the EGFR gene, confirming that these are indeed regulatory elements for EGFR. So all of what I just showed you in terms of the skewing was all done with publicly available data and not directly mapping the enhancers on amplicons, which we really wanted to do. So Andrew was able to acquire a cohort of about 40 human glioblastoma samples, do K27 acetylcholine ChIP-seq mapping, find the samples with EGFR amplifications, and then see if in fact you see peaks in those samples that have the actual amplifications. Here's what some of the data look like. Okay, so he was able to find, at the cohort of 40, he was able to find 9, where when you look at the input DNA from this ChIP-seq, you can find these large giant mesas when you align the data, indicative of a focal amplification at these regions. When you look at the actual ChIP-seq data, you can see that in every one of these amplified samples, there's the presence of the two enhancers that I showed you before. In addition, these enhancer peak sizes correlate with the copy number, suggesting that there's an enhancer on each individual copy of these circular DNAs. That certainly doesn't validate that these are circular extra chromosomal structures. So he followed this up with DNA FISH, where he took a probe to the EGFR gene, labeled it red, and probed metaphase spreads. So the EGFR gene normally resides on chromosome 7, which he lit up with a probe to the centromere of chromosome 7, the CEP7 probe in green. And you can see that all of the EGFR is not located on chromosome 7. It's virtually all extra chromosomal. Okay, it's all located outside of chromosomes. So these amplifications are in this sample are almost exclusively extra chromosomal. He was able to further mine the sequence reads from all of the ChIP-seq data and find these junction reads that support this circular structure. Okay, and in doing so, he's able to reconstruct what the actual DNA sequence of these structures look like. He can then layer on top of that what the epigenomic features are on these circles. And so that's exactly what he did. And so here's one tumor where he found this amplification. Let me walk you through what's going on here. Okay, so here's the input DNA. You can see these 3 giant amplifications. It turns out he's got junction reads or split reads that support that all 3 of these pieces are actually stitched together. Here's the epigenome across these regions. He's then able to reconstruct the circle in the context of all of the regulatory features. And this really blew us away when he did this, because we were expecting to see, okay, the EGFR enhancers are there. We were expecting to see this, but we weren't expecting to see this. All these other additional enhancers that they're sort of carried along on these amplicons are also included on the circle. It's just laden or rich with enhancer activity, okay? If you looked across other samples, you see the same thing. This is 3 additional samples that he was able to do circular reconstructions of in the context of their epigenomic features. You can see that in every one of these instances for the EGFR application, both the EGFR enhancer 1 and 2 are there. But beyond that, there's just a whole plethora of other enhancers. They're very heterogeneous from tumor to tumor. Okay, but the key point is that they're just riddled with enhancer activity. And so we scratched our head and said, what does this mean? What is that doing? Are these just passengers that are carried along? Does it function? Are these actually contributing to the expression of the EGFR oncogene? How can we start to tease this apart? So first he said, well, let's look at the 3D conformation of this circular structure. Okay, we know enhancers interact with genes through a physical interaction. He said, first, let's see, in the wild type situation, do we see a physical interaction with the EGFR enhancers and the EGFR gene? So what you're looking at here is high-C data. Okay, so this is data where you can map the 3-dimensional architecture, map interactions. He also did a technique called 4C-seq, which gives you even higher resolution to map chromatin interactions. Again, this is an EGFR unamplified glioblastoma sample. So what you're looking at here is, here's the locus, here's the EGFR gene, here's the 2 enhancers upstream, and he's finding that they are indeed physically looped to the promoter of EGFR, okay, as you'd expect. Here's the TAD, okay, so it's all contained within this TAD. So here's a rendition of what the structure looks like. At the base of the loops, there's 2 really nice CTCF sites. Okay, these usually reside at the base of the loops. They're thought to anchor loop extrusion to form these structures. We call this, for whatever reason, I don't know how this came about, but we call this the cheese curd structure. Looks like the cheese curd. We're in Wisconsin, so I thought that was appropriate. Okay, so here again is that same EGFR unamplified sample. And then here's what you see when you look at that amplified sample. So let me walk you through this. So this is those 3 segments. Okay, remember, these are all stitched together. They're loaded with enhancer activity. You can see that here from K27, the settle track. But when you anchor on the EGFR promoter, you now find that the regulation of the EGFR gene extends way beyond those 2 upstream enhancers to virtually every enhancer that was contained on the circle. Okay. And a lot of these even go beyond the TAD boundaries. Okay. So there's some sort of mode of unique gene regulation that's occurring on this circular structure. Okay. Something that's totally different than what you would expect to see given the structure on linear chromosomes. And it's suggesting that every one of these enhancers is really participating in the regulation of EGFR and boosting the expression of EGFR beyond what can be accounted for by the copy number alone. So coming back to the structure, here in normal you have the cheese curd, and in the tumor with these circular DNAs, again, you have multiple interactions with all of the regulatory elements that span TAD boundaries and blow through CTCF sites. Okay, are they functional? I know they're looping, but it doesn't tell us that they're actually functional. So Andrew, together with a technician, Zach Faber in the lab, designed a CRISPR-I screen. Okay, so what they wanted to do is they said, you know what, let's just go nuts on this. Let's march around the circle with guide RNAs. Okay, let's saturate the circle with as many guide RNAs as we can and use CRISPR to functionally test every enhancer. So they took 10,000 guide RNAs and a CRISPR dropout screen, and they tried to assess with sequencing which enhancers were functional, which ones contributed to an effect on cell proliferation or cell fitness. Okay, here's the structure again. Here's the unamplified. I'll walk you through this. Here's the interactions with enhancer 1 and enhancer 2. These peaks here, these pink peaks, correspond to the strength of that region as a contributor to proliferative fitness. So in other words, the height of these bars means that it's that enhancer or that region that was targeted had an effect on growth. So again, you see in the unamplified lines, a lot of these do in fact affect growth like you might expect, but also in this highly amplified line, virtually all of the enhancers, not every one, but almost every one of these seems to contribute to the overall proliferative fitness of these cells, telling us again that these are really functional, these are really required for these cells to maintain their high proliferative fitness. Let's get there. Okay, so everything I showed you is in GBM. Okay, and we wanted to know, is this a general trend? Okay, this idea that there's enhancer selection on these oncogene amplifications. And so Andrew was really a bioinformatics whiz, and he said there's tons of publicly available data out there that we can mine to try and see if this is actually a general trend. So he downloaded data, mostly from TCGA, from 9 different cancers spread across the body. He found all of the ones that were driven by focal amplifications of an oncogene. I forget how many he found at the end of the day, but there were hundreds of them. And over here, this is a busy slide, but over here are all the oncogenes that have a focal amplification. And he had ChIP-seq data to also mine from all of these cancers as well. So he could ask the question, how often when you have a focal amplification, is there an enhancer that's contained on there? And you can see that almost all the time, almost 100% of the time, these focal amplifications are not just amplifications of the oncogene. They're at least carrying along one additional enhancer on the amplicon. What's also cool is he can go back and address skewing. Okay, does he see these amplicons? They're skewed upstream or downstream. And this was really striking to us. He found some instances where the skew patterns were different for the same oncogene. So here's some examples of that. So here's EGFR with a 5' skew. I showed you that. Here's medulloblastoma where there's a 5' skew upstream of the MYC oncogene. And here's 2 different cancers, neuroblastoma and Wilms tumor, that have focal amplifications of the MYC-N oncogene. But the skew pattern is different. In neuroblastoma, the amplicons are skewed downstream, 3'. In Wilms, the amplicons are skewed upstream, 5'. And so he said, okay, this is great. Like, this is an opportunity for us to see, if in those regions with different skew patterns, are there enhancers that explain those skew patterns? Okay, so here's medulloblastoma. Remember, this had a 5' skew. Okay, sure enough, it's got an enhancer that sits right in that pattern of skew. Here's neuroblastoma. Okay, here's neuroblastoma with the 3' skew. It's a MYC amplification. Sure enough, in the region of 3' skew, there's this beautiful super enhancer sitting here. Here's Wilms tumor. Okay, it has a 5' skew. We didn't even have the tumor samples. He went to colleagues at the Cleveland Clinic and said, "Hey, can you get us some Wilms tumors? I want to profile them and I want to evaluate to see if there's this skew." And they said, "Yeah, sure. Here's some samples." They gave us samples. He profiled them. And we were just overjoyed to see that, sure enough, right in that region of 5' skew, you see this enhancer that's pulled along. And so that really kind of cinched it for us that there was this enhancer selection on these amplicons. He could further go and say, okay, well, what is can we use the enhancer maybe to see what the cell type of origin is for these cancers? So what he did for this was he downloaded data from the epigenomics roadmap where they have ChIP-seq across 150 different cell types. And he said, are there any cell types in the human body that have the enhancer located in the 5' or the 3' region of skew of the MYC-N gene? For neuroblastoma, the top hit was enhancers that are found in fetal adrenal. Okay, fetal adrenal is rich with neural crest cells that are known to be the cell type of origin for neuroblastoma. Okay, so that was really amazing to see. And in Wilms tumor, as you might expect, the 3' skew, or I forget where the skew was, but we're in the region of skew where that enhancer had the closest match to enhancers in embryonic kidney. Again, suggesting that these are the cell types from which these tumors are actually originating from. So to summarize this part of the talk, I think what we're seeing is that enhancers really do undergo selection on these amplicons and on ecDNA, and they probably shape the ecDNA amplifications. These enhancers engage the oncogene in a novel 3-dimensional conformation, one that's not seen on linear chromosomes. And these enhancers on ecDNA provide or endow a growth advantage to these tumor cells. And I think the reason why I find this interesting is because it really brings in, what we like to think of as a new concept or a new paradigm to this area. We've been thinking all along as the oncogene is the fundamental driver of cancer, and it is. I'm not saying it isn't, but I really think we need to start thinking about it in the context of all of the other regulatory elements that are brought along with it on these amplifications. And so I like to think of these structures not as an oncogene, but actually as like an oncolocus. Whole structure is important. You can target that structure, target the enhancers on that, kill cancer cells, okay? Or as the structure as a topological driver, okay? It's something new and different. It's got a novel topology in the cancer cell that that in itself is oncogenic. Okay, how does this happen? We're not exactly sure, but what we think, we think one model to explain it is, again, you have these in the cell type of origin, you have these chromosome shattering events, it's chromothripsis. In the situations where the enhancer is lost, there's no selective advantage, okay, for that cell to survive. There's a far better selective advantage when the regulation of those oncogenes is in fact preserved. Again, this field is red hot right now. Okay, this whole field of circular DNA. There were just 3 new papers that came out very recently. It turns out that these ecDNAs, first of all, they're far more prevalent in cancer than we had ever appreciated. Okay, we've known about these circular structures since the 1960s, but we've never been able to study them at this level of resolution. And we've also never been able to really evaluate how prevalent they are. It turns out they're common in as many as a third of all human cancers. It turns out there was this beautiful work by Chailin Wei and Roel Verhaak showing that there's crosstalk between these circular DNAs and the enhancers on them and enhancers on linear DNA. Okay, they bump into one another. Okay, the enhancers on the ecDNA can amplify the expression of genes on linear chromosomes. Howard Chang's group just, I think it was last week, published this Nature paper saying that these ecDNAs, these circular molecules, also form these hubs in the nucleus, and the enhancers on the ecDNA within the hubs talk to one another. Okay, so completely different, separate circles can interact with one another and influence the gene regulation on each of these circles. Okay, there's a million questions that remain. Okay, and in some ways, even though we've made tremendous headway, I feel like this cartoon kind of captures where we're at. Okay, we know there's something really fascinating in these discoveries, but we really haven't harnessed the power yet. Okay, we're sort of in the Stone Ages, if you will. There are a lot of questions. How heterogeneous are these circles in tumor cells? It's not an easy question to answer. Okay, these sequencing technologies we have are amazing, but it's still very hard to actually deduce the DNA sequence of these circles, partially because they're quite heterogeneous in these cells. And also, is there enhancer heterogeneity on these circles? And is this a basis of tumor evolvability? How is this novel topology established? Okay, and do the rules for circular DNAs differ from linear DNAs? Okay, we know that topology, that CTCF sites has a lot to do with how these connections are formed, but on a circular structure, it may be completely different. And lastly, it's just really spawned a whole field of whether or not you could target ecDNAs as a way to treat cancer. And can we target transcription on ecDNA, or can we target this novel topology or the regulators as one strategy that could have therapeutic benefit? So before I transition to like something that's completely new and different, I want to just ask if there are any questions or any concerns that I can address from anybody here about this.
Hi, I have a question.
You showed some IC data from normal cells.
Mhm. Yep.
If you perhaps you're seeing how it's also your
Mhm.
IC amplified.
Mhm.
You see now the EGFR enhancers bumping into enhancers and genes all over the genome.
So this is I haven't seen that in our high chip data. I think you have to, so the way Jalen and Roll did it, they had to normalize a special way to see that. We haven't done it the exact way they did it. And just looking at the data as it, as it comes off through a standard analysis, we don't see that. Okay. I'm not saying they're wrong. I'm just saying that we haven't seen evidence for that yet in that data. Any other questions? Yep.
So those amplicons, how they maintain when cells are divided? Are they still, so that some cancer has keep making those amplicons?
Yeah. So this is a fascinating question. I think what you're asking, correct me if I'm wrong, is like, how are these things replicated and how are they transmitted to their daughter cells? Okay. So these circular DNAs replicate once per cell cycle. Okay. But they're autonomously replicating structures. Okay. And in addition, what happens during cell division is they tether to the subtelomeric regions of chromosomes somehow. We know nothing about how this works, but they tether to the subtelomeric regions. And then as cytokinesis happens and the cell splits, wherever these things were sort of tethered and wherever these chromosomes get pulled apart determines how they're inherited and how many copies go to one daughter cell versus the other. It's completely random. Okay, it can be, if there were 100 copies, it could be 1090, it could be 2080, it could be 5050. And you see that when you quantify copy number in the distribution of a tumor population. You see this sort of Gaussian distribution, sort of telling us that it's probably somewhat random. Yep.
How much of an increase in the expression do you get from?
Yeah, I wish I had put the plots in. So what you see is if you plot copy number and then you were going to predict what the expression would be based on copy number, you'd see a point that falls right in the diagonal. In some instances, it's way off the diagonal. Okay, not in every instance though. Okay, so there are some instances where it's closer to the diagonal, there's some instances where it's below the diagonal. Okay, it can be in various places, but it's often much higher.
So is it a twofold increase in expression? Is it a 100-fold?
I wouldn't say it's 100-fold. It's variable, but it's more. I'd say it's between say 2 and probably 20-fold. Okay, and I think it's somewhat dependent on how many enhancers were carried along. But there's still so much that we don't know about what's accounting for that expression. It may in fact be Howard Chang's model of these things forming hubs and there being crosstalk among the enhancers. So there may not be a simple model to really explain why it's, when it's higher, why it's higher. And that's also why I can't give you a straight answer on it's because it's a range and it's so heterogeneous between these tumors. Yep.
You probably can't do this experiment, but can you test some of those models? Can you add in extrachromosomal circular DNA and have a promoter mutation and then see whether the enhancers on one side are active?
It's a really hard experiment to do for a couple of reasons. These are giant structures, they're megabase sized structures. You can't really reproduce them by putting plasmids in, okay? So the field is wrestling with the best way to model these things. One way is you can sort of make them by making cells methotrexate resistant. DHFR locus during the evolution of methotrexate resistant will form a circular DNA. Another way to make these, which is still very much an experimental phase, is you can cut with 2 guide RNAs and the fragment that gets liberated from the chromosome will actually circularize. Okay, so we're actually working with the group that's developed that technology to do those exact experiments. And we want to mutate the crap out of the regions, right? We want to mutate the hell out of the loci, the enhancers, the promoters, everything. Just put in a bunch of mutations with CRISPR to try and get at the very questions that I think you're asking. Yep. 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 Yeah, how does it work? Yeah, so it's a Cas crab, decast crab construct. So it's a Cas dead version, okay, hooked up to a crab repressor. Okay, and when these were designed, it was thought that they probably work by like actually like repressing any sort of activity there, actually like silencing, but they probably work through like occluding the site and preventing transcription factors from binding. It's not really clear, honestly. But what we're doing is we're targeting the whole region, and then just sequencing the output to see what dropped out, what guide RNAs dropped out. So it's a CRISPR dropout screen. So from that you can infer which regions were functional.
Yeah, I actually took part, but I worked with how, so the gene is actually fast.
Yes.
Does it have to, like, is it have to see like this, that the
I see. I don't know. We didn't do that experiment. It's a great experiment that should be done. Like I think what you're asking about is it actually affecting the contact? Yes, we don't know. We don't know. I suspect it is. I suspect it's somehow breaking the interaction, or at least mitigating it in some sort of way, but we don't know. And I think we need to follow that up, because I think that we took a very transcriptionally centric view on it, that it was just preventing the transcription factors from binding. But the other possibility is that it's somehow interfering with the replication of these circular structures. Okay, origins of replication are often contained within DNA enhancer elements. Okay, so origins double as enhancers in a lot of cases. So it's very possible that what we're seeing is not only due to a transcriptional effect, but by inhibiting the replication of these structures. So we'd like to tease that out. We haven't thought of a really crafty way to do it yet, though. Okay, I'm going to move on to something completely out there. Okay, so if I'm just sort of trying this out. This is a newer story in a field that I think really deserves a lot of attention these days, and that is the field of addiction, and in particular addiction to opioids. So there's the whole world is in a crisis right now, and there's a massive raging opioid epidemic. In 2017 alone, there were more than 100,000 opioid overdoses. Okay, and in 2017, it became the leading cause of accidental deaths, exceeding automobile accidents. And this crisis is only worsened with COVID. I mean, it's just been devastating. Now, we know that opioid use disorder is 60 to 80% heritable. That is a huge genetic contribution. Okay, that's on par with other more common traits, if you will, things like schizophrenia. Height is, I think, 60 to 80% heritable. Okay, so there's a huge heritability component to this. Okay, it runs in families. Okay, but it's been very hard for the community to figure out where the heritability is, what genes actually account for this. Okay, so GWAS so far has uncovered one replicable hit, and that's in the opioid receptor, mu receptor gene. And it's been thought that we're going to get there. We will get there as a community, but it's going to take hundreds of thousands of samples to really uncover additional risk loci. And this probably reflects the complexity of this disease and the heterogeneity of this disease and all the factors that go into the complex trait of opioid addiction or addiction to anything. And so we ask, can we accelerate this process somehow? Can we shortcut the discovery of genes involved in the neurobiology of opioid use disorder? And so I helped form a consortia with the groups that are shown here. And we collectively reasoned that maybe we could do this. Okay, and I'm not claiming that we have solved this problem by any means. Okay, but I want to show you some new data that I think is pretty enticing and encouraging. Okay, so we reasoned that there were probably some key features of a successful approach. So first, we reasoned, since a lot of times these GWAS SNPs are localized, that confer predisposition to disease, or localizing to enhancer elements. Okay, we reasoned that we needed accurate measure of epigenomic activity and connectivity to genes. Okay, so we needed to be able to figure out where are the enhancers, where are the genes. Of course, we need to do this in the appropriate cell type. Okay, that's difficult with diseases like addiction. Okay, you probably need parts of the brain, right? And so we got that, but you get the point. And then also we need to think about approaches that kind of consider that this is an incredibly heterogeneous disease. Okay, we're not going to find one spot in the genome that explains everything. Okay, we have to somehow embrace the heterogeneity. All right, so the experimental design was rather straightforward from the start. The idea was to start mapping the enhancer landscape in dorsolateral prefrontal cortex of humans, and we were fortunate enough to have a collaborator, Deborah Mash in Miami, who's been banking brains from folks who have overdosed from opiates, okay, mostly heroin. And they all come with toxicology reports, so we know if there's any other drugs in the system. I think for 90% of the cases we have, there were no other drugs, purely heroin that led to their overdose. And then we also have a cohort of accidental death controls. These are mostly car accidents. And so what we did was H3K27 acetyl ChIP-seq, so mapping of the active enhancer promoter landscape across this cohort. We had some promoter capture high C data that was publicly available that we could use to connect enhancers to the genes. And we also did, so we didn't do that on 51 samples or whatever, we just had some reference data. And then we also did RNA-seq studies. And we also did this on neuronal nuclei sorted from the postmortem brains of these individuals. Okay, so it's ChIP-seq, postmortem brain neurons. All right, so what do we find? So the first thing we noticed is hold on a second. There's just like this. There's clearly this effect of global hypoacetylation that we saw. Okay, so here's an example of just a subset of the data. I think this is like 20 of the controls, 20 of the cases. Here is one region where you see this subtle but clearly present difference in acetylation at the stealth-4 locus. Okay, but by and large, there's this global effect, this hypoacetylation effect that we had seen. But the effect is really kind of wimpy. It's really kind of weak. It's like 1.15 fold. It survives statistical significance, no problem, but it's really kind of wimpy. And there's only out of tens of thousands of peaks, there was 388 significant peaks that we retrieved here. So this data is useful. I'm not trying to be dismissive of it. It's telling us that there's this chronic use of opiates is probably having a pretty global effect on acetylation of the brain. Okay, but we reasoned that there's probably more to the story than that. And how can we leverage what we've done before to try and tease out more from this? Again, the effect sizes are weak. So we started looking for VELs, very much like we've done in cancer, gains and losses of enhancer activity. Now everything's hypoacetylated, so you can forget about gains for this disorder. All the action is going to be in the losses. But essentially what we did is we looked for instances where you have a lost VEL or a lost enhancer activity in each individual case relative to the whole cohort of controls. Sorry, back up. And when you do that, you can see that these VELs have much more of a stronger effect size than we saw with the traditional linear regression analysis. And we also saw that there's, as may be expected because there's global hypoacetylation, this preponderance of these losses, okay, nearly 81,000 of these case-specific losses that we saw. So then the story got interesting because we said, well, what the hell do we do with 81,000 losses and how do we start to make sense of this? So we started linking all these to the genes, okay? And we did this with the promoter capture high C data. And we noticed that first of all, the number of VELs, the number of these losses that you find in 2 or more cases is very small, okay? Meaning that they look like they're kind of scattered around the genome. But when you link them to the genes, you find that most of them, like 60% of them, are actually linked to a common gene. So it's telling us that there's some convergence, there's hot spots in the epigenome where you have this, you have these changes occurring. So it's coming back to this dimmer switch analogy. If you have 3 enhancers, you may have patient 1 has a lost VEL at one enhancer 1, patient 2 has one at enhancer 2, patient 3 has one at enhancer 3, right? So there's heterogeneity across the patients, not the same site, but it's the same regulatory circuitry that's being perturbed across the cohort. Okay, and so we call this convergence analysis. Okay, we're looking for instances where the circuitry of the gene, all the enhancers in the gene, or the plexus of the gene is another word we use, shows perturbances, and you're looking for this. So you're looking for statistical evidence of this. And there are a variety of scenarios. You can find instances where a single patient may have multiple enhancers for a gene messed up. Here's an instance where you have a single patient or single element that's perturbed. But the most common are these situations where you have multiple enhancers for the plexus disrupted across multiple separate patients. And when you run stringent statistical analyses to look for this, what we found were 5 genes, okay, listed here: ASTN2, KCN, DUSP4, so on and so forth. Okay, so these 5 genes, we only found 5 genes, okay, but nonetheless, we found 5 genes. Okay? We found 4 more than OPRM1, okay? So again, at this point, it's all just statistical support that there's something going on at these loci. And here's some examples of this. Here's that DUSP4 gene that I showed you in the beginning. Again, this is a gene that actually has a single element. It's a rare gene that has a single element perturbed across multiple patients. 19 out of 51 cases have at least one. But by and large, we see this erosion that's occurring at the plexus or at the regulatory circuitry of this gene across the cohort. Here's one of the top hits, this ASTN2 gene. It's got a ton of interactions and regulatory elements that control its expression. And there's just massive erosion of these enhancers across the cohort. So of 106 elements for this gene, 69 of them are VELs, and 11 out of 51 cases have at least one. And we started mining the literature. Okay, what do you do with 5 genes? How do you start to demonstrate that these actually have anything to do with opioid addiction? And our top hit, there was a paper that came out right around the time we were analyzing this data that said, okay, that there was a GWAS study. It said AS2 and ASTN2 polymorphisms are associated with patient-controlled dosing of opioid-based analgesics. Okay, so what the heck does that mean? What that means, this was a study where they took post-operative patients who had the ability to hit the morphine button and self-administer opiates. And they found that the top hit in their study for the people who pushed the button more was ASTN2, okay? So again, this really interesting circumstantial link to this top gene that we're finding associated with opioid overdose. So this got us thinking, well, let's take these genes and let's see if there's any additional GWAS or any additional genetic evidence that would support their role in opioid abuse. So we took these 5 genes. It's just 5 genes. It's not a lot of real estate across the genome. And we did an approach called LD score regression, where we look for traits where there's genetic evidence for genetic support for these regions. And we cross-reference our regions with a whole bunch of different diseases shown here. The top 3 hits were years of education, number of sexual partners, and generalized anxiety disorder, okay? And the enrichment just for these 5 regions was way higher than expected, even for the enhancer landscape alone, okay? There's really strong, striking enrichment for these 3 traits. Now, it turns out that these 3 traits, it's hard to know what to make of this, okay? And there's I'm not by any means saying years of education determines whether you're going to die of opiates. It has nothing to do with it. What it's thought of is that some of these traits, particularly years of education, number of sexual partners, these are surrogates for risky behaviors and also for early life adversity events, okay? So in the field of psychiatry, these are surrogates for that. And they're known risk factors for opioid abuse. We further took the GWAS study, okay, the one that had the 1 hit at OPRM1, and we just asked the question, are the regions that we're seeing, do they show any overlap with these sort of lower subthreshold hits? And sure enough, whether you mine the top 1%, 0.5%, or 0.25%, you find that there is enrichment, there is statistical enrichment among these regions. So I'm running out of time here, but I think what I'm getting at is that we're seeing that there's this epigenetic variation in these opioid overdose cases. These are converging on these genetic vulnerabilities with known risk factors for opioid abuse. And we're arriving at these opioid use candidate genes. And it's the takeaways from this is, we've uncovered 5 genes here with a really small cohort, nowhere near what's required for these GWAS studies to find these hits. And we think this convergence framework that was designed can be used to augment or accelerate the mapping of these complex genetic architectures. Okay, what I'm trying to say here is in addition to these GWAS studies, start looking at the chromatin, start going right to where the action is, and we think you'll find relevant biology. And at this point, we're at the tip of the iceberg here. We've just found 5 genes, but we've only looked at 50 cases and 50 controls. But nonetheless, we believe we're capturing these shards. We describe them as shards of the genetic and the environmental factors that are perpetuating this opioid epidemic. And of course, the next question is what actually are we uncovering? And are these actually pre-existing changes, or are they genetic or environmental, or are they due to exposure? Okay, and so we're trying to think about ways in which you address this. It's very complex, hard work, but I think we're on to something. At least the genetics seems to be supporting that. So I'm going to stop there and thank folks from my lab who were involved in the study, and I think I've thanked other people along the way. So thank you for your attention, and I'd be happy to take any additional questions.
That was really interesting. I'm just wondering, the 5 genes that you got out of them, do they show differential expression between the 2? Maybe I missed that.
Yeah, so some of them do and some of them don't. So DUSP4, the one that has the most erosion, clearly shows a difference in expression. The other 4, honestly, we're underpowered with the RNA-seq data to see it. It may not be there, but I think we're actually underpowered, because what you have to do is you have to take the 11 or 12 cases that have the valve, match them up to the 11 or 12 cases from the RNA-seq that have them, okay? And unfortunately, we actually don't have RNA-seq on all 50 cases and controls. We only have a subset of those. So I think we're actually underpowered. But yeah, it's an obvious question, and we need to figure that one out. Anything else?
All right, let's thank Peter one more time.